Thanks very much to the MPA committee for giving me the opportunity to speak about this area. So today I'll be talking to you about Duchenne and Becker muscular dystrophies, and hopefully I'll demonstrate to you why these aren't just muscle disorders, but are actually brain disorders that we should be giving some more neuropsychiatric attention to. So just firstly, I'll just give a brief kind of clinical background to Duchenne and Becker muscular dystrophy and talk a little bit about what we know about dystrophin brain involvement and current research, and then I'll present the real-world database study that we performed. So just giving you some background about Duchenne and Becker muscular dystrophy. They are X-linked recessive dystrophinopathies, so they predominantly affect males. And in the UK, they're very rare. We estimate that we've got about 2,500 males living in the UK with Duchenne currently. And they're involved in the dystrophin gene, which is one of the largest genes in the human genome, which codes for the dystrophin protein, and that acts as a kind of shock absorber that wraps around the muscle fibers and protects it from damage over the long term. And without the dystrophin protein, you get progressive muscle fiber damage that leads to kind of replacement with fibrosis and fatty tissue over time. And what we see clinically is that males with Duchenne typically have delayed motor milestones from infancy, and then in their early childhood years they might present as more clumsy, falling over more, not as able to keep up with their peers, and then their diagnosis is normally between the ages of kind of 2 and 5. And then by teenage years, they lose the ability to independently walk and require wheelchair assistance. And over their 20s, they get progressive respiratory and cardiac compromise, which is the life-limiting element of this disease. Even given the current treatments that we have to prolong life expectancy, which are steroids, cardioprotective treatment, and then invasive ventilation later on in life. And what we know about the genetics of the two disorders, so Duchenne is thought of as an in-frame, out-of-frame mutation, which leads to completely absent or aberrant production of the dystrophin protein, whereas Becker is thought of as an in-frame mutation, leading to a kind of milder phenotype with a later age of onset and slower progression of the neuromuscular decline. But these two conditions are kind of thought of as a spectrum, and there is variability in the phenotype that we see. So just talking about the relevance of dystrophin in not only in the muscle but in the brain, we've come to understand that both the full-length dystrophin protein and also shorter isoforms that are transcribed from promoters along the gene region have been found to be implicated in brain activity. So DP427, which is the long protein, has been found to be expressed in both the cortex, the hippocampus, and the amygdala, and the smaller isoforms DP140 and DP71 have been implicated in preclinical studies in neuronal migration and synaptogenesis in the fetus, and also in GABAergic and glutamatergic signaling, which gives us an indication that this might involve kind of EI imbalance, and that's been kind of borne out in mouse models where you've had dystrophin knockouts that show that mice demonstrate increased startle response, changes in their executive functioning and planning, and it's also been borne out in fMRI studies showing increased excitability or increased activity of the default mode network in males with Duchenne. But we have limitations in the amount of data we have clinically because it's such a rare disease. And the largest cohort studies that we've got to date is this 2022 systematic review and meta-analysis from Pasquale Moreno et al. And they brought together 23 cohort studies with over 3,000 participants from international kind of clinically ascertained cohorts. And these, they brought together prevalence estimates for a number of different neuropsychiatric conditions. And you can see at the end here, the mean prevalence estimate. So you might notice a really significant estimation of rates of ADHD, both in the Duchenne and in the Becker muscular dystrophy population, with estimation of rates of kind of up to 1 1 in 5 or even fewer with being diagnosed with ADHD, but also high rates of anxiety, about 1 in 4, and also, increased rates of ASD, depression, and OCD. But we wanted to really kind of drill down onto the neuropsychiatric associations that we could see in Duchenne muscular dystrophy using a kind of larger cohort. So we leveraged the TriNet-X database. You've heard a little bit more about that previously, but this is a kind of international cohort of from over 18 countries with, I think, over 200 million participants now. So you can leverage their anonymized electronic healthcare records to create different cohorts and compare outcomes between groups. So we created the dystrophinopathy cohort, and this was patients that had ICD coding for either Becker and Duchenne muscular dystrophy, and this was actually combined in the ICD coding. So these are kind of dystrophinopathies at large, not differentiated between the two groups. And we compared them with healthcare controls, so people that came for a routine healthcare visit with no associated diagnosis, but also wanted to look at another progressive neuromuscular disease, so compared with a cohort that had been diagnosed with limb-girdle muscular dystrophy, which is another progressive neuromuscular disease causing eventual disability and is a life-limiting disorder, but is more genetically heterogeneous and doesn't involve dystrophinopathy itself. And then we also compared to female carriers, so they have they're haploinsufficient, so they have one normal X chromosome, and they're normally asymptomatic, although they sometimes do display kind of some degree of muscle weakness and have a high propensity to cardiomyopathy, but associations with neuropsychiatric disorders hasn't really been well evidenced in literature thus far. We propensity score match to adjust for confounders such as age, sex, race, and socioeconomic status. And you can see the outcomes that we were looking at that were also based on ICD coding within the TriNet-X database. So this is just a slide kind of comparing the TriNet-X cohort, which you'll see in red, to the previous systematic review data. And you'll see that there are comparable kind of estimates of prevalence in autism, in anxiety and depression, but notably kind of lower prevalence estimates of ADHD and OCD in our TriNet-X cohort. And also we included other neurocognitive disorders, including intellectual disability, language disorder, learning difficulties. And you can see very low rates of severe mental illness, such as mania, bipolar, and schizophrenia. And then these are just odds ratios looking at comparisons between the rates of diagnosis with all of the neuropsychiatric outcomes that I mentioned before. So this is comparing dystrophinopathy patients to the healthcare controls. And you can see a 5-fold increase of likelihood of being diagnosed with an intellectual disability, but also much higher rates of diagnosis of autism, OCD, and learning difficulties. The keen-eyed of you will see that there was no significantly increased risk of diagnosis of ADHD, and that was a really surprising finding. Part of that may be explained by the fact that the healthcare controls and a large proportion of TriNet-X cohort is coming from the US, which may explain why there's higher rates of baseline ADHD diagnoses. But you'll see when we compared to the limb-girdle group, and also I'll just go to the next one, to the female carriers, there was a signal found for increased rates of ADHD diagnosis. But you'll see that it was, we could still see these effects in the limb-girdle group, so comparing to another neuromuscular illness, these kind of increased rates of neuropsychiatric disorder can't really be explained just by virtue of having a chronic neuromuscular illness alone, and we still see kind of high rates of all of the neuropsychiatric disorders mentioned before, and also this is reflected in comparisons between the DMD, BMD males and female carriers. And just the last piece of analysis that we did was looking at rates of neuropsychiatric illness in female carriers, and you can see that there was a slightly increased signal in rates of diagnosis of both autism and language disorder, which may suggest that there is an implication of even kind of partially reduced function of dystrophin in the brain in this female carrier group. So just to summarise the findings, we did see that there was this a distinct profile of neuropsychiatric comorbidity in the Duchenne and Becker males, and it is this kind of spiky profile that clinically you do see a kind of overlap between intellectual disability and ASD and ADHD, but these TRINET cohorts may be underestimating the rates of ADHD diagnoses in this group, may be due to diagnostic overshadowing and the limitation in access to specialist ADHD assessments. But we also see that there's this intermediate phenotype in female carriers as well. I won't spend too much time talking about the limitations. I think one of the main things that we were limited by is that we couldn't do molecular genetic confirmation of the DMD or BMD diagnosis, and we were reliant on ICD coding instead. And it would be very useful to kind of drill down into the genotype-phenotype correlations, looking specifically at the impact of the isomer differences that I mentioned before. But I hope that this has just highlighted how holistic care in this rare neuromuscular group is essential, and early screening really can have a very significant difference in someone's long-term psychiatric well-being. And as we move towards the very promising future therapies such as gene modification therapies that have been instrumental in prolonging life and improving the neuromuscular symptoms, we need to be thinking more about the impact of neuropsychiatric disorders on these patients, and we want to kind of improve quality of life as well as quantity of life. And as I mentioned, future research that can be done. I just want to thank everyone I work with in the Pollack Lab, in KCL, and in the neuropsychiatry clinic that I was previously working with, seeing these Duchenne patients in Queen Square, and all of the members of the psychosocial working group that work in DMD Care UK. And this is just highlighting the recently published psychosocial standards of care guidelines for both mental health professionals and also neuromuscular consultants from that charity. Thank you.
Thank you, Talia. Time for one question.
Can I pick you up on your control group of limb-girdle muscular dystrophy?
Yes, please do, yeah.
I mean, that would be comparable to your Becker's group, but not to your Duchenne group.
Yeah.
Duchenne are far more severe. Schooling, social participation. So matching for diagnosis isn't enough. You need to match for disability. You need to match for, what effect it has on school, age of onset. So, so I would expect to see a dissociation between the Duchenne and Becker group, and limb-girdle isn't a good control group for Duchenne.
I take your point, and thank you. No, it's definitely a limitation. We struggled to kind of think of a genetically kind of based
spinal muscular atrophy.
Yeah, I know. I might extend the analysis and include the spinal muscular atrophy group as well. It's a very good point. I know it's a kind of more clinically heterogeneous group, but that doesn't have as severe progression of their neuromuscular problems. So yeah, it's a good point. Thank you.